Thank you for coming, everybody. Today I'll talk about discovering coding sequences using transformer networks, um, which is not the most hard to understand title that I've ever heard. But let's go forward. Nope. All right. Oh, great. So maybe the more important question is, why am I the one to talk about this? I'm a Belgian native. You can maybe notice that from my family name, but I was born in Belgium. I was there until 2.5 years ago, and I also studied there in Ghent University. I did a bioengineering master's, a PhD, and a little bit of a postdoc there. I did some freelance with Novo Nordisk, and during my PhD, I'll first focus on my PhD. During my PhD, I focused mostly on processing biological sequences using deep learning techniques. This is, 4 to 8 years ago. So back then, this incorporated a lot of novel stuff like convolutional neural networks. Multi-layer perceptrons was still seen as pretty new. Recurrent neural networks, those are kind of out of fashion at this point. And then the end of my doctorate was more about transformers. That was kind of the new thing, and I was quick to jump on it. And I was mostly working on prokaryotes, which was great because we didn't have the biggest computational resources at the university. So it allowed me to kind of work with a smaller data set. As part of my postdoc, I worked with Novo Nordisk also through my freelance. It's a pharmaceutical company, as most of you will know. And they were mostly interested in, therapeutical targets. And then I moved my focus on the human genome, and specifically trying to find, translation products that have not been annotated yet. Now my postdoc ended. My wife is American. I had worked with John Prenzner before. He was just starting a new lab here, and that's how I ended up at the University of Michigan 2 years ago. So all of this research is kind of part of a bigger subject, which is referred to as the dark genome, which we basically just kind of means stuff that is being transcribed and translated that is not yet known. We have our canonical coding sequences, we have our canonical transcripts, but more and more research sees that, especially in cancer types, you have all these things that are happening caused by mutations, caused by oncogenes. And the interest is just, okay, maybe part of what is happening wrong can be used and leveraged to use as a therapeutic target. If we understand this better, we could understand that maybe part of these novel translation products are drivers or can be used for immunotherapy. I think DMG is a very good example, diffuse midline glioma, where a mutation on the histone gene causes a lot of the DNA to be accessible. So a lot of these cancer cells are driven by a lot of translation and a lot of transcription. This has become more and more of a hot topic, I would say, or maybe I'm a little bit biased because I'm focused on it. And this is also a lot what John Prenzner, as part of the Prenzner lab, what we're focused on. You see this more in research. There's been lots of articles which specifically target things that are referred to as non-canonical operating frames, micropeptide, novel translons. There's lots of different jargon, which is maybe somewhat confusing, and we'll get to that in a second. But it's definitely something that's living. You also see it in the press, etc. There's concerted efforts from multiple labs across the world, including our lab, to create public databases using all kinds of methods that have been in existence for a while, using all kinds of data, and make this accessible to the public so also they can include this in their research, just to make sure they're not missing something. This is stuff that is in collaboration with GenCode specifically, which is also where John Much is affiliated with. And you can find this list. I think recently there was a new version of this list. But this is just to say, this includes a lot of labs, and it's a big concerted effort. Not a newer paper, which also Christian has worked on, specifically looks on the peptide type of the side, where we first predicted these to be in existence, and then we want to do mass spec and all kinds of analysis on the peptide level to ensure or validate a lot of these proteins. All right, so translation products, non-canonical operating frames, translons. It's a little bit confusing, and I think there's a little bit of a history where things have been maybe misnomed, because and technically a open reading frame, let's try to use this pointer, see how it goes. An open reading frame at the top right is technically is anything in between a valid start and a valid stop codon. So if you're talking about all possible open reading frames, a transcriptome, it just means anything that is between an ATG and a valid stop codon. An open reading frame does not necessarily mean being translated. And also, unfortunately, not all coding sequences, which we can refer to as annotated translation products, exist within an open reading frame, because sometimes we have things happening such as non-canonical start sites, start codons. We have frame shifts. We have things where the ribosome ignores the stop codon. So they're not necessarily on an open reading frame. So a recent paper, which is basically from a couple of months ago, or maybe even this month, introduces this concept called a translon, which just means a translation product. And I also probably refer to this if I say something like a non-canonical open reading frame or a micropeptide. I think it's basically that. So how can we detect novel translons? That's the big question that I was asked 4 years ago, I think, about. And I chose to, well, the first thing I did was, okay, let's focus on sequence context. I think the most obvious thing to focus on. And then what I specifically thought is, okay, they come from RNA transcripts, so let's look specifically at RNA sequences. Why would it be useful for me to try to use machine learning, since it's my background, to try to apply machine learning to solve this thing that has already been tackled or has been worked on for many decades by big consortia with all kinds of techniques and validation, all types of orthogonal data types. There's maybe two reasons. I think the most one is this is something that has existed for a long time. So there's some assumptions that have been made over time, and these assumptions propagate through data, versions, new releases. I think two very important ones are listed at the bottom. You have these multi-coding sequence transcripts, which means if you ever worked with a GTF file or an annotation file, every transcript has a maximum of one coding sequence. Something that is kind of known to not be the case, but that's what GTF supports, and GTF is the thing that everybody uses. You also have the non-canonical start codons, so if it's not ATG, it's generally not an ensemble or GenCode. So you want to try to see whether, yes, the data, the current annotations are pretty perfect or not. We don't know. The idea is machine learning is known if it has 5 to 10% noise to still be able to optimize well, and then we can see if it predicts novel things. Maybe that's the stuff that gets missed by the big consortiums like Ensemble and GenCode. Transformer network. I guess I could have put a lot of slides of which network are we going to use, but we live in modern times, and at this point, this question has been reduced to let's just use a transformer network. I mean, this was a big question because things go fast, but 2 years ago, or actually 4 years ago now, this was not a given. But transformer networks are great, and I love them, and I've used them for a long time now. They have this thing called automated feature extraction, which is something that deep learning shares. It's not a specific thing to transformer networks, which just means that you don't have to manually start annotating, okay, which part of this RNA transcript is relevant to have a coding sequence. Like an ATG is important. Let's give it a higher weight in our machine learning, or let's make it a feature that is used as input to the machine learning. We can go a step back and just give it, this is all the information we have. a full transcript sequence, you figure out what's important here. This is big, especially in biological, in molecular biology. Most of the data we work with is pretty obnoxious. Well, obnoxious is not the right word, but obtuse. So there's also great parts where it's just efficient. Maybe before, where we would use a convolutional neural network, you have to choose like a specific input region. If you want to make a prediction on site 1, you want to have 100 nucleotides upstream, 100 nucleotides downstream, give that as input, make a prediction on that site. If you want to then make a prediction on the site next to that, you've got to, again, input up to 100 nucleotide sequence, but then shift it by one. So you're just reprocessing the same inputs over and over again. With transformer networks, you don't have to do that. You can just give the RNA. I think I'm just going to use this. I'm going to just give the RNA sequence at the bottom, and then it makes predictions on every position for every nucleotide or transcript. You don't have to at once. So it parallelizes that. It also uses GPU, but again, shared by deep learning. Also kind of a big deal. And of course, state-of-the-art performances. And it also handles label imbalance well, which is something which I'm not going to talk too much about. It's an interesting topic. But oftentimes in machine learning, when you have a problem where you have 1,000 times more negative samples than positive samples, it's sometimes really hard to optimize your model. All right, experimental design. So what is the learning problem? We want to identify these, novel translons. How can we do this? Well, translon is in effect an area, which means that you have to create some output where it starts and it stops. But this is kind of very, very complex because how do you combine the two? So to just make things easier, what the model actually does is it predicts where the translation initiation site is. And then we just do a greedy search until we find the first in-frame stop codon. But you can say, not all coding sequences live in open reading frames. This is true. Unfortunately, we're not going to be able to pick those up. And that's fine, because if we want to take into account all possible start stops with possible frame shifts where there's possible stop codon read-through, then the problem would just be infeasible to optimize. This is, in essence, a binary classification task. I am unfortunately not 100% sure on the background of machine learning of most of you. I hope this is somewhat easy to understand. If it's not, I'm okay with questions in between the presentation, but I'm not sure what the habits are here. I'll just move on. So if you look at the data, and we want to do this for the human genome, we're talking about 260,000 transcripts. We're talking about 440 million nucleotide sites on each transcript. And then the positive set is, I think, 1/10,000th of that, or 1/55,000th of that. So very imbalanced data. Then when we optimize this and we try to select a model with our validation set and then benchmark our performance with our test set, we make sure that all our transcripts come from the same chromosomes, because there's something called a transcript isoform, which means very many versions of the same gene, but then transcribed in very many different ways. And of course, our performances might be biased if we have a very similar transcript in the training set, and then we're validating or benchmarking our test set. All right. So to then cover the full human transcriptome, we have to train, unfortunately, 5 models, because well, if you want to predict on our test set, then we cannot use it for training. So we have to do this 5 times. Great. This model is not super big. It's surprisingly small. It's about 120,000 weights. This is a small language model, which you don't read much about, I guess. But it's very successful. So this is kind of what that looks like, right? So we get our validation loss, which is, while we train, we monitor our validation loss over time. Epochs means you go through your training set fully once. Then your loss is determined by your loss curve. your loss function through which you do optimization, gradient descent. Again, I can't go through everything based on your background. I hope kind of following. But basically, the lower the loss function, the better. The better the model approaches what is seen as good answers. We can also measure this as with the precision recall area under the curve. And what you typically do is you go to minimal validation loss and then you evaluate on the test set. So this is for the 5 models. We see that if we take different folds over chromosomes, these performances are very similar, which is great. We try different transformer sizes, and we also see we have the transformer medium, which just calls the transformer here. And then we have the transformer large in green. No, it's the same, yeah. And we see that the large doesn't really improve much, so we just take the medium so people don't have to wait 5 days to finish training. And the output distribution looks something like this. If we look at our negative set, that just means those 440 million nucleotides. And then the positive set means there's 80,000 translation initiation sites. We can see that those are, and this is on our test set, right? We can see that these are pretty much pushed apart, which is excellent. Because that means, and we can also see this reflected in the precision recall curve, this means that our model actually is able to optimize very well and do a pretty good job at determining where our translation initiation sites are. Whoop, I jumped. All right, we did some benchmarking. Maybe it's not super interesting, but it performed a lot better than what was already in existence without needing necessarily more parameters, meaning model weights in this context, nor needing more training time. But then, of course, the ROC curve and the PR precision recall curve are a lot better. So good results, happy. Next step, want to see like, okay, what's going on? What is interesting, I guess, is if you then look at how our model performs on existing annotations, since that's what it's optimized with, we can see that the performance is a lot higher for transcripts where there's more support. And support level, transcript support level is one of those terms that is kind of an aggregate for the more support we have through orthogonal means, data means, the more sure we are of this transcript actually existing, and the better. And the more in this case means a lower number. So 1 means more support. So in general, we see if a transcript is part of the coding sequence, consensus coding sequence set, if it has a high support, then our model performs really well. If we're dealing with transcripts that have basically no annotations and low support, then it's kind of performing worse, which can imply that maybe our input data is just not good. So it's not our model's fault. I mean, I'm biased in my own way. That's how I like to interpret these results, but that's how I do it. We can do something else where we look specifically at the false positives. So say we take a threshold here at this line, we can say that some of our negative set is predicted as positive. What if we specifically look at those false positive predictions? And then we can do a p-blend on them. For example, has it been found in UniProt in some form? And then we get, of course, more matches. Then it's just a sanity check, but we have more matches again where our model output is higher and our support is higher. So all these results are pretty good. So we're pretty happy with that. This kind of looks like that. If you want to look at, you want to plot it per transcript. Again, we have a prediction for every position. So this blue line is basically all those predictions next to each other. And then sometimes you see something jumping out. That's just a higher prediction. Like obviously, since we have 400,000 nucleotides, most of those predictions are basically 0. Sometimes we have a big jump. The darker ones are the canonical coding sequences. The light blue ones are the novel translons. And we have all kinds of different things that we can see, right? We see the downstream open reading frames. We see the ones that are upstream, meaning upstream of the canonical coding sequence, etc. Sometimes we have an internal extension, which just means the stop site is the same, but we have a start site that is more upstream of the canonical version. These are very well-known terms if you ever get your hands on literature that specifically looks into this. The upstream open reading frames, the long non-coding RNA open reading frames, the downstream overlapping open reading frames. That's what we're talking about. Great. So step 1, excellent. Pretty happy with that. Can we do more? We can, because there's something called ribosome profiling. How am I doing on time? Oh, actually taking longer than expected, as always. What is ribosome profiling? I'm sure not everybody is familiar with that term. It's one of the things we do at our lab. So ribosome profiling is a lot like RNA sequencing, with the difference that in ribosome profiling, you specifically filter out the fragments that are protected by your ribosome. So if you specifically take out those fragments and you map them, then instead of getting a signal all across your transcript, you get a signal specifically across your coding sequence, which is great because we're specifically interested in coding sequences and not transcripts. The signal looks a little bit wonky or like periodic. I'm not sure how you say that, but it looks like this because, of course, when you do translation, your ribosome reads codons, so triplet nucleotides, and that's how you get this type of signal. Now, how? Yeah. Now, there's this thing where you have multiple different reading lengths. Sorry, I will go back actually. So if this is your read, your ribosome read, and this is your ribosome where it exists in, sometimes you have, in your ribosome profiling, you have read lengths of different lengths. Sometimes they're 28 nucleotides, sometimes they're 30 nucleotides. And based on that length, you can have a different offset on how the 5% end of that read, this one, positions itself as with respect to the E side in your ribosome. So typically you want to, or what is historically done is you calculate offsets for each ribosome length, and then you get a mapping signal on your genome, on whatever you map to. Not ideally, if you look at these fragment sizes, what I was just saying, then you look in which frame do they exist within your coding sequence. If you then apply that offset, right, that I was saying, so we apply an offset to map it to one site, one nucleotide site on your transcript, we can see that for different read lengths, they are positioned differently in your reading frame. On top of that, there's the problem where depending on whether you're working on autopsy samples, cell lines, tissue samples, depending on the organ you're working with, depending on the translation inhibitor you're using, and depending on just the lab protocol, that profile or signature looks completely different. This is just 3 different data sets where, again, we have our different lengths and just how they kind of map with the colors to have a clear vision of which reading frame it is. And again, we see it's just completely different for different samples. On top of that, and I don't have a figure for this, but it's also not a given that this profile looks exactly the same for every gene. We'll come back to that later. First, we're going to talk again about our ribosome experimental design. It's basically the same thing as with this transformer, with the only difference that we're using ribosome profiling data instead of sequencing data. So, and also, of course, what the model does is different. If you use your sequences, you're basically saying we're re-annotating the genome. Any of these coding sequences or translons could be translated at any point. When you're using ribosome profiling data, you're obviously saying this is being translated in this specific sample. So in that way, they're also slightly different. But the data set, the binary classification is the same. It's just the input embedding that's going to be different. So part of the study was, okay, how do we look at this? What can we do? There's multiple things we can do. There's the strategy A, where we specifically look back at what has been done historically is to calculate an offset for each read length. You just kind of apply that offset and get your read count for every position, which is displayed there. Alternatively, we can use strategy B, where we are just gonna not do anything. We're gonna keep our reads like they are. We just take the 5% end of our read. We're gonna take fractions, and then we're gonna do something very fancy with the input embedding. Without going too much into it, how can I explain this? Basically, in the initial approach, we start with a continuous input between 0 and 1, because we do normalize this. And then we want to get an input embedding from that. So I send it through a multi-layer perceptron, do a tangent hyperbolicus transformation on it, which means that I get a vector with values in between -1 and 1. Then we use that as some type of mask for an embedding that is optimized during the training process. So this is directly optimized, meaning that every value of your array gets optimized through gradient descent, going up and down with each training step. And then this mask is what you apply on top of that pre-optimized, optimized embedding during when this model gets applied. This is using strategy A. Alternatively, we can also sum that with some kind of aggregate embedding of your reads, where you just basically do a summation of your individual embeddings, where each embedding is just optimized during training, and each embedding belongs to a specific read length. So those are those fractions. You basically multiply your fraction with your embedding, you sum them, you get your EL, which is referred to as here, and then you sum that with your EC, which I reference here. I hope everybody can see that, because that's maybe a little bit low for some people. How did I come to that? I have intuition and a lot of machine learning and stuff they do in machine learning. But it works. I did all kinds of things too, but not everything is, of course, included in the paper and is worth talking about. But this was a solution that worked well. We tried this for different data sets. We did the A strategy versus the B strategy, where with the A strategy, we can take the naive 5% position, or we can use specific methods that calculate these offsets for specific read lengths. Didn't really matter. Strategy B won. So we went with strategy B. And in essence, you basically don't lose information, whereas when you apply strategy A, you do lose information because you're going to apply the same offset for every gene, transcript. Whereas with strategy B, how these read lengths are positioned with respect to each other, you keep that information because it is different with different transcripts. Part of the optimization process is also, okay, this is great. I trained it, took a day or two, not great, because I do want this to be useful for people that are not me and that have time to apply this to lots of samples. So something else I looked into was pre-training, which is also a very popular thing. You can think of it as transfer learning, where I just kind of pre-train a model, and then when you apply it on your personal set, your specific ribosome sample, you do some fine-tuning, meaning you optimize it a little bit on your sample just to get those offsets that specific reads have right for that specific sample, and get your predictions afterwards. This is what I do here. I do just supervised pre-training, meaning I just train a model on a lot of samples, and then when you apply it on another sample, you train it again, but you start with the weight set that you get at the end of your pre-training, which generally results in an optimization time that's way faster. I also did something self-supervised pre-training, which is something that is often done with these large language models. The same thing, if you think about language models, you don't need labels. And large language models, how that looks like is typically you just take Wikipedia, you send in a sentence, but you mask some of the words, and you let the machine learning model predict which word it is. Now, automatically, if you do something like that, the model is going to learn something about how language is structured. And that makes a lot of sense because there is a lot of structure there. And that's a good way where, without defining a specific objective, you can already create a model that is very powerful. And once you have that model, you can apply it to a lot of different tasks without having to retrain a whole model and learn how language works from scratch. So this was successful. The supervised pre-training was the most successful technique. So that's what I stuck with. And our optimization times were about 45 minutes, which is very reasonable. The benchmark was, again, very good. It worked better than other tools. It had about 50% increased retrieval of coding sequences than the second best tool. And then specifically with small peptides or small coding sequences, it had about 300% more. So very happy again. We also had some other interesting results, such as when you do ribosome profiling, and you get a profile like this. Sometimes you get a profile like the left thing, and sometimes you get a profile like the right thing. The right thing is considered excellent. The higher your in-frame read fraction as compared to your out-of-frame read fraction, the better the model is, because historically the tools used to work a lot better on those. Now, if you look at the results from Rabotai, which is how I call the tool, we could see that when we look at in-frame read occupancy and then the performance, there was a slight correlation, but it was not significant. The correlation that is significant and very relevant, of course, is your read depth, which is very cool because now we can use low quality samples. All right, what did we learn from this? Small large language models are effective, which is fun because I don't know, I don't read many studies that use those. Now, of course, large language models are ideal for processing variable length inputs, which used to be a big problem. We used to have recurrent neural networks for that, but those are just so slow. You don't want to deal with those. So transformers are great. Maybe I should call them SLMs instead of small large language models. That doesn't make much sense. Apologies. We got our two models, this transformer and Rabotai. They do different things. They have different biases. So they're used kind of together as orthogonal ways of validating whether we have a translon or a translon of interest. Because this transformers worked is trained on the sequence data. Well, since all the canonical coding sequences, or the majority, 99 point something% start with ATG, then obviously the model is also gonna score all ATG starting open reading frames a higher score. Rabotai doesn't have this issue because it only uses ribosome profiling data. It doesn't use any sequence data. So it doesn't have those potential biases. It does require fine-tuning, which is not ideal if you don't have a GPU, and it does make the tool less accessible to anybody. But it does work very well. So is that the end of the story? Unfortunately not. Well, unfortunately, things are more complex than that. This is the tool, or this is kind of an overview of the tool. It's kind of a complex thing at this point. I'm not sure what I want to say about this, but typically you don't need much. You can do your database creation with your FOSTA and your GTF file. If you want to do Rabotai, you need your BOM file, but otherwise you can just apply this tool. For example, if you have applied string tie, you got some not interesting novel transcripts, you can just go to the GitHub. It has pre-trained models for both humans and mice. So you don't have to do this problematic training step, because that does take, you have to train 5 models, so that can take a week, depending on how many GPUs you have, at least. But yes, most people, well, not most, but a lot of people work in human data, so that is accessible. If you're interested in ribosome profiling data, well, our lab specializes in that. So maybe just reach out to the lab. Unfortunately, also, the story's not quite done. You have something called coding sequence variance. You can think of it like with ribosome, and this is a problem specifically to maybe Rabotai or the use of ribosome profiling. But your ribosome map to the genome, and then you map them to the transcripts. We have something called transcript isoforms. So obviously, if a ribosome read maps to a specific transcript that is part of a coding sequence, which is kind of like your canonical transcript, part of the gene, the transcript that's generally expressed in a gene. Now, if you ever used Ensemble, you'll see that a lot of genes have 20 transcript isoforms. So if you run Rabotai, you might get some, like a lot of novel translons. It's great. There's upstream of reading frames everywhere. But unfortunately, oftentimes it's just the reads that map to those, that other transcript that is kind of mapped on the same region on the genome can also map to all those different transcript isoforms. So when you get a signature, a ribosome profiling signature for a specific gene, that signature will be shared by all those isoforms. So oftentimes we get predictions on those isoforms where we think, it's probably not an upstream open reading frame, probably just you know, this annotated coding sequence that lives on the same genomic region on another transcript. Fun, more complications. Good thing is that I spent a lot of time in the last 2 years dealing with that. So this is also part of Tristan as a tool set. And this is too small for you guys to read, obviously, but what is actually shown is just the different types of open reading frames and then the result before filtering and after filtering. And I think you can see that the right-hand bar is a lot lower than the left-hand bar. So it's an important thing to take into account. So what do we do next? We try to validate them. And now I'm going to talk a little bit more about what we do at the lab and what we do with this information. So a lot of these translons we can send through, of course, foundation models, which is great because if you send them through these foundation models, you get embeddings back. You can try to UMAP cluster them. You can do supervised UMAP too. And then these are good ways to kind of separate out the likely false positive as compared to the actual really interesting candidates. And we have different models to do this with. So this gives us, again, different types of information. ESM and AlphaFold are by Meta and Google DeepMind. They're more focused on the protein's structure. So those embeddings will have more information about protein structures, whereas EVO2 is more trained on a ton of genomes from a ton of different organisms. So that will give you more information about whether sequences are more preserved throughout, and just kind of the structure on nucleotide level or genome level, transcript level, whatever. I'm also working on supporting mutations. We're working in cancer, so obviously mutations are a big part of that, which is also a lot of fun. More complexity. So part of our pipeline now is kind of, we have our RNA-seq data. We use string type to infer more transcripts. And then we also use deep variant, which is also a DeepMind tool to use RNA-seq data and predict where our mutations happen. Then using our mapped ribosomes, profiling data, and using these mutation data, I'm expanding our set of predictions with even more predictions, including potential novel open reading frames that get created just by mutations happening on those transcripts. Stuff we do at the Pranzer Lab includes lots of types of things. And we're all trying to bring this together. So we have a RNA-seq and a Ribosome, which creates expression profiling. We can also look at translational efficiency, which is a unique aspect of having both. We're currently also working with RNA methylation. So obviously that can also have an effect. We're just looking at regulation, all different factors of regulation and how can they affect expression in our organisms. So our mutation signatures come in, our methylation signatures come in, our translation, like our Ribosome data comes in, and of course our novel open reading frames or translons, come in. Trying to combine all of that together to figure out what is going on in these cancers. That's the end of my presentation. I want to, obviously, this work was not possible with the help of a lot of people. Pranzer Lab and also Biobix Lab, which was my lab at the University of Ghent University, and then also with Novo Nordisk. Of course, a lot of the work is done by our wet lab people and our dry lab people. Some of us, well, most of us are joining us today. They're doing amazing work. Obviously, there's a lot of work that is done before somebody like me gets my FOSQ file, I would say. So thank you to all people involved. And we're ever growing, we're doing really good. So if you have any interest, please reach out. Thank you. Questions?
Thank you for your time. I'm just curious about your positioner encoder design. So unlike other natural languages, I think the order of input sequence of genome may be somewhat hard to design. I mean, how do you design the position encoder for this transformer?
The position encoder. Yes, I use the rotary embeddings. Does that make sense?
Well, Is there any other consideration behind that?
This is a good question. This is a question that I would have been better at answering maybe 2 years ago, because I've been focused a lot on the bioinformatics side. No, I think, I mean, if you look back at, how do I go back? For this transformer, I used something called absolute embeddings, which obviously was not the best because rotary embeddings didn't exist anymore. Actually, I don't think it's in here, so I'll just go back to the end. No, I just use the rotary embeddings. I've thought about that at some point for a week, but I've stuck with that for now. I feel like that's still to be considered.
Yeah, it's still a problem. Yeah.
A problem, yes, but it's also considered to be one of the better ones, right?
Yeah.
Okay. Unfortunately, I can't say more. Yes.
This is entirely new to me, so I guess I could say that I find your translon concept disturbing.
Oh, well, it's not my paper, luckily.
But how do you know, for example, that if you do your ribosome profiling or you're binding transcripts to ribosomes, these transcripts generally will have a 3D profile, 3D shape.
Right, yes.
And those 3D shapes may have different configurations that are more prevalent than others.
Yes.
So each one then could bind to the ribosome in a different place or
yes.
way.
Yes.
So you may be sort of overextending the amount of so-called translons that you think you add.
No, this is, yeah.
I mean, what's your comment on that? I mean, is that something you can correct?
Yes. So the question is whether the transcript shape, the structure of transcripts themselves, they must have an effect on the translation because of how the ribosome can approach the transcript. So what are my thoughts on that? Yes. No, this is very real. And the thoughts are this: with ribosome profiling, I don't have to worry about it because the ribosome reads give proof of those ribosomes being there. So the ribosome profiling is kind of proof of expression happening. With this transformer, where I only look at the sequence data, it's possible that the sequence data is not enough to make that to have enough information to calculate whether the structure of the RNA is going to have an effect and therefore result in a lot of false positives. And I agree with that. One aspect that is interesting is the model performs very well, which is one thing. And part of the paper, we also did some integrated gradient something study where we look at which inputs are of importance to make a prediction. We did see that even though we give the transformer, we only give it a sequence, we do see that based on those studies that the model has learned where the stop sites are, has learned where the codons are based on just seeing the importance. We see peaks at where the stop site is, the stop codon, which we never told the model of where it is. So in that same fashion, it is likely that the transformer has learned other aspects that is of importance to what constitutes a valid coding sequence and what doesn't. But it is a black box, so I can't say for certain whether something has been learned about the structure or not.
Thank you.
This is why having two orthogonal approaches is very useful.